Hi everybody, welcome back. So we're going to continue to explore this idea of sex-linked inheritance and specifically understanding um, genetic differences that can ultimately be traced back to differences in these XX and XY chromosomes. So genes can be located on either of the sex chromosomes, but daughters and sons don't necessarily share the same probability of inheriting those traits. So for example, you can have something called an X-linked trait. And an X-linked trait is simply a phenotype. Again, what we're looking at is the expression of this gene that is determined by an allele on the X chromosome. So an example of a recessive X-linked trait basically is a normal copy of one X chromosome, and it masks the recessive disease allele on the other X chromosome. So a primary example of this is looking at a specific type of muscular dystrophy called DMD. So your book does a really great job of explaining and understanding how this inheritance of this X-linked recessive trait can be passed on and affect males. So here we have two different images that are looking at different scenarios that can happen in terms of inheriting this X chromosome trait. So for example, here we have the father who is unaffected, carries the XY chromosome, and then we have a carrier mother. So remember that the normal X chromosome is masking this disease in X. So when we do this sort of Punnett square and start looking at what the reactions can be, we see that we have one unaffected daughter who is not a carrier. We have an unaffected daughter that is a carrier, again, where that X dominant gene is masking that disease on the X chromosome. And then we can also have an unaffected son, or we have an affected son, because here there is not another X chromosome to overshadow that recessive X allele. Now over here we have another very similar chart, but in this case the father already has DMD, so again has that X chromosome that has that affected allele, and when you compare it to somebody who has an XX, you can see that you have two unaffected daughters and unaffected sons. But again, both the daughters are always going to carry that recessive X allele. So what are the takeaways of looking at this X-linked trait? So the first thing that we have to always understand is the mother passes one of her X chromosomes to her children. However, the father passes only the X chromosome to his daughters and only the Y chromosome to his sons. So because of this difference between, quote, mothers and fathers, or those that have X chromosomes and those who have XY chromosomes, we see that boys, or in this case, those who have XY chromosomes, have a higher probability of inheriting X-linked diseases than girls, because again, a girl will, in this case, quote unquote girl, has two X chromosomes where one X could potentially mask potential errors on the other X chromosome. And a way that we can look at this in understanding different inheritance patterns, we use a chart called a pedigree analysis to really understand how inheritance patterns might affect different families. So a typical pedigree chart might look like this. Again, this is just sort of a visual representation of the occurrence of different phenotypes across generations. So you start up at your oldest generation, where you have in circles detailing those that have XX chromosomes, and squares detailing those who have XY chromosomes. And again, here in red, we see that that X is going to be those that have that DMD disease, where the normal X chromosome is overshadowing that inherited X chromosome. And we can see how that X is then inherited across this this tree. So you see here that you have an aunt that again is a carrier, and we know she's a carrier because your cousin has the actual disease expression of DMD. So we know then that that aunt had to get that that second red X chromosome from the original, in this case, the grandmother. On the other hand, for example, we have two cousins, one that is XY and one that is XX. But in this case, because we don't have an individual that has the disease representation, we don't know for certain if this is an individual who has an XX or an X with that inherited DMD mutation. Your book, in this case, also looks at another example of a pedigree analysis, looking specifically at the generations of Thomas Jefferson and Sally Hemings. And I put a really great resource page that I'd like you to explore, which is a museum exhibit looking at the life of Sally Hemings. And they go specifically into this genetic analysis that your book briefly covered. But this is a great visual art piece that recently came out this last year, where the sixth removed grandson of Thomas Jefferson was photographed using the same clothing as Thomas Jefferson. And it's a really powerful art piece that again is highlighted in that museum exhibit page. But how exactly did we start to understand whether or not Thomas Jefferson had children or fathered children with Sally Hemings? And the answer is we're going to be looking at this Y chromosome analysis. And the important thing and way that we can really understand the Y chromosome analysis is because it is a shorter chromosome, there are really only a few genes that you need to look at. And because there are fewer genes, it's a really great way to sort of understand, study ancestry, and identify paternity. But why does it actually have fewer genes? Why can we rely on this Y chromosome? And one of the primary reasons we can really rely on Y chromosome analysis is this concept called recombination. And recombination is essentially chromosome crossover that can occur between pairs of genes during metaphase of mitosis or meiosis. And you can see here we have two genes that are next to each other, and they're able to actually switch segments of DNA and genes that happen across these chromosomes. So it's essentially a way of intermixing and adding genetic diversity within the cell environment without introducing mutation. So what does that mean when we're talking about the Y chromosome? Well, because sons always inherit that Y chromosome from their fathers, we're able to sort of compare the DNA sequences, but you can see here that there's no recombination possible between the X chromosome and the Y chromosome. So where X and X chromosomes are able to recombine, able to increase that genetic diversity, that is not true when we're looking at the XY chromosomes. So because of this, we know that we can really use that Y chromosome and know that the types of genetic diversity that we're seeing are directly related to the father and not because of some sort of crossing over or recombination that's occurring in the X chromosomes. So because of this sort of lack of genetic diversity, we're able to compare again those short tandem repeats, which you'll remember from your DNA and PCR lecture that we covered. And we're able to actually create and recreate these genetic histories. And because of that, we were able to create this published journal that in 1998, which was covered in your book, that is able to link Thomas Jefferson with male line descendants and Sally Hemings. So that really covers what we're going to be going through in terms of understanding sex-linked characteristics. And in our next lecture, we're going to start to understand other types of genetic inheritance that are possible outside the world of specifically sex-related diseases and lineages. Okay, see you soon.